And this is exosomal ACE2 neutralizing broad variants of virus SARS-CoV-2. So the world has entered the third year of the COVID-19 pandemic, and the virus is by no means gone. Still over, as of April 2022, over 497 million confirmed cases, over 6 million deaths, and only almost 11 million vaccine doses administered. And what really exacerbates the whole situation is the appearance of mutants of the virus that have higher infection rate, higher transmission rate, higher mortality rate, and thus they are posing a great challenge to the existing treatment and vaccination. Thus, there is an urgent need to develop therapeutics against broad strains of the virus in order to save human lives, especially those susceptible people that are immunocompromised, those that are not yet vaccinated, with a low vaccination rate worldwide. And of course, with the appearance of more virulent variants every now and then. So the common thing about the wild type and all the variants that appeared is that the virus, through its spike protein, it binds to angiotensin converting enzyme, as we all know, on the cell membrane of the host cells. This is the way it can get the door that it uses to get into the cells and infects it. So if there is a way that we can just close this door, inhibit this binding, then we have inhibited the universal mechanism of infection. And we have developed a therapeutic against broad strains of the virus. So with this concept in mind, and starting from the fact that the convalescent plasma from COVID-19 patients have been used to treat patients with a severe disease, we aimed to identify new antiviral components from the human plasma to yield potential new therapeutics. So we used microflow cytometry or microflow vesiculometry, and using this nice technique, I'm trying to show my cursor. Yeah, and using this nice technique, we were able to find that the levels of the ACE2 expressing extracellular vesicles in the human plasma of acute phase and convalescent patients, okay, is significantly higher than those of the seronegative, which are healthy donors. Also, the levels of these vesicles, again, ACE2 expressing vesicles, is higher in inpatients as compared to outpatients. We confirmed that the levels of the plasma ACE2 extracellular vesicles are associated with the severity of the disease. Nice. So what is extracellular vesicles? Let's refresh the memory. So extracellular vesicles are shed. These are, as the name implies, these are vesicles that are shed by the cell in order to communicate with its surrounding environment. They can come in a range of sizes. They can be apoptotic bodies, bigger ones up to from 800 to 5,000 nanometer. They can be exosomes up to 200 nanometer, or they can be microvesicles, the smaller ones, which are up to 1,000 nanometer. From this, we have exosomes have been regarded as the best characterized small extracellular vesicles that likely participate in a variety of physiological and pathophysiological functions, as well as serve as a novel biomarker. As you see here, they are loaded by interesting cargo for communication, such as proteins, mRNA, and they have very significant markers on their surface, such as CD81 and CD63. So starting from endocytosis of proteins and RNA, these endocytic vesicles are delivered to the early endosomes, which fuse with each other and result in the formation of late endosomes or multivesicular bodies. These bodies either release the exosomes by fusion to the cellular membrane, or their contents are degraded if they fuse with the lysosomes. Actually, exosomes are now regarded as a unique drug delivery vehicle. This is because, as you see here, therapeutic proteins or sensitive molecules such as DNA, RNA can be loaded inside those exosomes. Other molecules such as hydrophobic drugs, adhesive proteins, hydrophilic drugs can be bound to the surface of exosomes. Actually, targeting ligands can also be found, and thus rendering exosomes as a targeted drug delivery system or platform. This platform has three key characteristics that give them an edge over existing platforms. They are less immunogenic, okay? So they are particularly useful for repeated drug dosing. They are less toxic. They are more able to cross the blood-brain barrier. So from our findings with the patient plasma and from what we know about exosomes, we developed our interesting hypothesis, which is SARS-CoV-2 infection enhances the secretion of ACE2 positive extracellular vesicles as an innate antiviral response. And actually what supported our hypothesis is our initial finding using ACE2 overexpressing lung epithelial cells. We found that viral infection enhanced the expression of ACE2 expressing vesicles after 72 hours of infection compared to non-infected cells. Okay, let's test our hypothesis. We need tools, and that's why we use two different cell lines, HEC and HeLa, that are engineered to overexpress human ACE2. We isolated extracellular vesicles from these cell lines using ultracentrifugation, and it's worth mentioning that yes, we end up with an extravesicular pellet of extracellular vesicles, but it is highly enriched in exosomes. We characterized those vesicles using nanosite. We also found that using cryo-EM, we found that ACE2 expression was detected on the IVs isolated from ACE2 expressing cells, but not from those isolated from the control wild type cells, while the extravesicular marker or the exosomal marker CD81 was expressed in both types of vesicles. Also using microflow cytometry, we found that within the extravesicular vesicles expressing the exosomal marker CD63 or CD81, we detected ACE2 vesicles, ACE2 expressing vesicles using this nice technology. And finally, using the conventional Western blotting, we found that expression of ACE2 was associated with the vesicles that are isolated from or derived from ACE2 positive cells, and they are associated with exosomal markers, which was not the case in the vesicles isolated from the control ACE2 control ACE2 negative cells. Their vesicles expressed no ACE2. So now we have a very nice model. Let's start the functional assays. So we started with cell-based assay in which we have developed labeled spike receptor binding domain. We labeled it with streptavidin, and we incubated it with ACE2 positive cells. So the RBD is going to bind to the cells and label them, and so they can be detected by flow cytometry, as we see here. However, if we pre-incubate the RBD, the labeled RBD with ACE2 expressing vesicles, the RBD will bind to the vesicles, and there will be less of the RBD found to bind to the cells when we incubate the mixture with the cells. And by flow cytometry, we found inhibited events for the labeled ACE2 positive cells. So with this nice cell-based assay, we were able to find that those ACE2 positive vesicles from the HEC or the HeLa inhibited RBD binding to ACE2 positive cells in a dose-dependent manner. And in fact, when we compared them to soluble recombinant human ACE2, they were 135-fold more potent. We were very excited about this result, and so we wanted to confirm it with another model. And that's why we used the pseudovirus. We used a pseudovirus labeled with luciferase reporter. We pre-incubated it with ACE2 positive or ACE2 negative control vesicles. And then we added the mixtures to expressing ACE2 cells. After 72 hours, we detected the luciferase activity, and you can see here that ACE2 vesicles in green prevent pseudoviral tethering and infection of the human host cells in the human host cells in a dose-dependent manner, which was not the case with the control ACE2 negative vesicles. And when we tested our vesicles in a dose-dependent manner, we found that ACE2 positive vesicles, both types of vesicles, did show this neutralization of viral infectivity, and they were from 30 to 60-fold more potent than the vesicle-free soluble recombinant human ACE2. And then at that time, the variants started to appear. So we tested the variants, and voila, we see that the same inhibition in a dose-dependent manner. At that time, we had alpha, beta, and delta. And actually, ACE2 positive vesicles neutralizes virus variants with similar to better efficacy compared to the wild type. So we had the nice cell-based assay, we had pseudovirus, and so we wanted to find out if this is true with the live virus. But before I move to this, I would have to confirm that. With the pseudovirus, we wanted to confirm that our vesicles did not sensitize the pseudovirus infection to ACE2 negative cells. And you can see here in blue, these are the ACE2 negative cells, and in green are ACE2 positive cells. And with or without the vesicles, the virus infects ACE2 positive cells only. This was something important that we needed to confirm. And so after this, we moved to the live virus. And so we incubated live spike virus with, again, our vesicles, whether ACE2 positive or the negative controls. And we incubated the mixtures with 0.6 cells, which express ACE2. And then we performed a viability assay where we detected cell death as an indication of live virus infectivity. You can see here that the cell death is enhanced, is decreased in a dose-dependent manner, indicating the decrease in a live virus infectivity, which was not the case with the control ACE2 negative vesicles or exosomes. Again, we compared our vesicles to recombinant human ACE2, the vesicle-free soluble recombinant human ACE2, and we get 90-fold more potency with the vesicles compared to the soluble protein. So we detected the variants, and we can find here that yes, the live variants are neutralized. The infection of the live variants is neutralized using our exosomes, and we confirmed in alpha and beta variants. Thus, ACE2 positive vesicles neutralize broad strains of live SARS-CoV-2 virus. And then it was the time to detect this in the animals. And so we used transgenic mice that are expressing human ACE2. And we incubated the live virus with our exosomes and then injected them intranasally to the mice, intranasal injection. We monitored the mice for 6 days. We looked at survival, viral load analysis, and pathology. And here are our results. So the mice that were administered ACE2 positive vesicles showed higher severe disease-free survival compared to the mice that received the control vesicles, which are ACE2 negative, and the results were very significant. The viral load with the mice in the lungs of the mice receiving ACE2 positive vesicles was lower than that observed in the mice receiving control extracellular vesicles that did not have ACE2. Looking at the pathology of the lungs, we found that ACE2 positive vesicles significantly inhibited acute as well as chronic inflammation in the mice of the lungs compared to the control vesicles that were ACE2 negative. We could observe also inhibition in alveolar hemorrhage and necrosis. So we had an observation. That there is initial observation enhancement in ACE2 expressing extracellular vesicles in the plasma of patients, wanted to find what is the function of this ACE2 positive. We confirmed the result in a cell-based assay, pseudovirus assay, live virus, as well as with animals. So let's find out if the function of these vesicles, as we have characterized them in our functional studies, is the same in the plasma. So we went again back to the plasma. And we isolated extracellular vesicles from the plasma using ultracentrifugation. We had acute cases, convalescent cases, and here I'm showing you the results of 2 patients of the acute cases. You can see here that the extracellular vesicles of this acute patient are expressing ACE2, and actually most of the patients, or we can say all the patients that we have detected, acute patients, had ACE2 and extracellular vesicles. Only 1 patient that we did not find detectable expression of ACE2 in the isolated vesicles. So with these isolated vesicles from the plasma of the patients, we did the same assay that I mentioned before, where we incubated the live spike virus with the vesicles and then added them to ACE2 expressing cells and looked at the viability of the cells. You can see here that ACE2 positive expressing vesicles enhanced cell viability as an indication of inhibition of infection with the virus. Okay, this was not the case with the seronegative or the healthy donors, where their extracellular vesicles were ACE2 negative, and this was not the case as well in this single patient that we have found not expressing ACE2. So this was a very exciting result that confirmed our finding, and we can now say that the circulating ACE2 vesicles from a human plasma neutralize SARS-CoV-2 infection. We wanted more confirmation, and this is why we further, after isolating the plasma extracellular vesicles, we further depleted ACE2 from these isolated vesicles from the pellets using RBD-bound beads. And mass spectroscopy analysis of the RBD bead pull-down materials from the patient plasma EVs pellet confirmed the presence of ACE2 and EV protein. We looked at the level, the counts of ACE2 and those EVs. We can find that there is significant inhibition in the levels of ACE2 positive EVs in the depleted samples compared to their non-depleted counterparts. And so we performed a cell-based assay, simple cell-based assay, and you can see here that plasma EVs with ACE2 without any depletion, they decreased the RBD binding to the cells. However, bead-depleted samples rescued RBD binding significantly compared to the respective non-depleted samples. This is another confirmation that circulating ACE2 positive vesicles from human plasma neutralizes SARS-CoV-2 infection. And one final thing that we did is that we looked at the expression of RBD IgGs in the plasma of convalescent and acute patients. There is enhanced RBD IgG expression, and actually the elevated RBD IgG levels in COVID-19 patient plasma are significantly associated with their neutralization effects on RBD binding to the human cells. Combining ACE2 positive EVs with the factor of RBD IgG levels significantly improved the model fitting to explain the RBD neutralization activity of the plasma. So these results again confirmed the antiviral contribution of circulating ACE2 positive vesicles. So we speculate the following 2 potential mechanisms as underlying how EV-ACE2 possibly achieves a better efficacy than soluble ACE2 to block SARS-CoV-2 infection. The first one is, as a small vesicles, they are an average of 100 to 200 nanometer. The proteins presented on those small vesicles might amplify the space intervals and thus suppress SARS-CoV-2 access to its host cell surface. Another hypothesis is that EV expression, extracellular vesicle expression, may increase the affinity of ACE2 positive binding with the virus protein through synergy among the ACE2 proteins on the same vesicle. And also the transmembrane domain, which is involved in presenting ACE2, it might be presenting optimal ACE2 conformation for binding with the S protein compared to the soluble form of the protein, which is globular protein structure that might be not showing the receptor, the binding domains efficiently as compared to those of the exosomal ACE2 or vesicular ACE2. So with this, I can just, again, these hypotheses need further future studies in order to confirm or find out what's really happening. And now we can summarize our findings. So the virus uses cellular ACE2 to get into the cell and infected. EVAC2 or ACE2 expressed on the extracellular vesicles can bind to the virus. And we believe that it might, those vesicles with the virus are swimming in the circulation until they are engulfed by immune cells and attacked and resolved by immune cells. And so we have produced, and the features of extracellular vesicles ACE2 are, they are a plasma biomarker associated with COVID-19 severity.
Lamia, you have 2 minutes.
Okay, they have high efficacy compared to soluble ACE2. They can neutralize broad strains of the virus, and they can be delivered intranasally to the lungs and airways, and they have good biosafety, low toxicity, and low immunogenicity, and they can be used as a biological platform for broad therapeutic development. With this, I would like to acknowledge the lab members in Dr. Weeping Lu Lab, our collaborators, especially Dr. Dewey Feng at Northwestern, the flow cytometry core at Northwestern and other cores such as pathology core, the BSL-3 facility at University of Chicago, and of course, our major collaborator, Dr. Raghu Kalluri at MD Anderson. With this, I end my talk, and I'm very happy to take any questions.
Well, thank you, Lamia, for a wonderful talk, beautiful presentation. And the talk is now open for questions. People can either raise their hand or type them into the Q&A. I have a quick question. Can you elaborate a little bit more on the mechanism that you think is going on? Is it simply a matter of the ACE2 expressing vesicles soaking up a sufficient amount of virus to prevent an infection, or is there something else going on? And if it's the former, when you expect that the more vesicles you give, the better, and is there a way you can test this mechanistically?
So your first question, you want me to elaborate about what you think is happening? That
yes.
Exosomal ACE2 better than the soluble, right?
No, just the whole idea of giving the ACE2. I understand your method has improved the delivery of the ACE2, but is it really just a question of giving the host enough receptor to soak up the virus and prevent it from infecting cells? Is it, in other words, it's just competing for viral binding, if you will, to with the cells, with the host cells?
Yes, the answer to your question would be, now those vesicles are used as a decoy.
Yeah, okay.
Okay, so they are decoy capturing the virus, and the virus binds to them efficiently, and it's not available to bind to the cells and infect them. It's a decoy.
And is there any idea of how much you need to deliver? And it would seem that this would vary a lot depending on the host level of virus and so on and so forth.
Absolutely. So with the animal experiment, we used a large amount, because it was a proof of concept experiment. It was used about 130 micrograms. Now we have very good funding to move forward with this project and produce more, do different settings of the treatment. So we know now that it's prophylactic. So we have more settings and we're doing more tests. Mm-hmm. And part of this is to do GMP production of high amount of exosomes that are expressing higher amount of ACE2. Okay, and in this case, we would increase the content of ACE2 per micrograms of exosomes. And so we would be injecting less of the exosomes and however, pertaining the same amount of efficacy. We are trying to do this now.
And I may have missed it, but are you altering the affinity of the wild type ACE2 receptor to be more high affinity for the virus?
No, we didn't do anything. It's the same as it is.
Eric, did you have a question? Or is your hand?
Hi, Lamiaa. Yeah, I have a couple of questions. They're all with respect to the animal model or the animal studies. I'm sure you answered this and I just missed it. Can you clarify whether the extracellular vesicles that you're using in that study, are they synthetic? You're made using recombinant proteins and synthetic lipids? And then the other two questions are, did the timing of delivery matter with respect to infection outcome, if you took a look at that? And the last question is, once you inject these vesicles into the animal, I guess intranasally, how long are they effective for?
Okay, the first question, whether these are synthetic exosomes. No, they are not. So they are cell-derived vesicles. So we grew the culture in a huge amount of cells and isolated the supernatant and did ultracentrifugation, and these are the vesicles. They were isolated from HEK cells. I made sure that we are not using cancer cells. So these were isolated from HEK cells overexpressing ACE2. So the vesicles were overexpressing ACE2. So this is question number one. Question number two, the timing of the delivery. This is a very important question. Thank you for bringing it up. What we did was an experiment of a proof of concept. We just did what we did with the cell culture. So we incubated the virus with the vesicles and then introduced it to the animal. It was again a proof of concept. Now we have other plans. We can say that our results imply or reveal that there is a prophylaxis. Okay, prophylaxis from infection, but we have a question mark. What about the infected animals? If we inject them with the exosomal ACE2, are they going to treat or not? So this is another setting. I hope this answered your question about the timing of delivery. The third question, it was to inject, I don't remember. It was to inject.
The third question is, if you just inject or put the vesicles in the animal, how long do they hang around for? So if you think of this as some way of preventing infection, can you inject a person once a week or something like this?
Again,
is what I'm driving at, yeah.
So what is the route of injection? We have now confirmed the intranasal injection, right? So if we look at the intranasal injection, when we did this experiment, I did a biodistribution experiment, and I found that after 24 hours, I can detect the exosomes in the lungs, but none of the other organs. It wasn't detectable in the liver, spleen, kidney, any of the other organs. So I can say that up to 24 hours, I could find significant amount of those exosomes in the lung after intranasal injection from my biodistribution. I have nothing to say beyond this because now we are in the phase to study the pharmacokinetics of these exosomes in the body of the animals using different types or different routes of injection, intravenous, intranasal, IP, within certain periods of time, prolonged period of time in different organs, by large biodistribution studies.